proceedings paper. The title of the paper is Onto2Vec, Joint Vector-Based Representation of Biological Entities and Their Ontology-Based Annotations, and will be presented by Fatima Zohra.
Hello everyone, my name is Fatima Zohra Smaili and I am a PhD student at CAF. So today I will be presenting our work on producing vector representations of biological entities from ontologies and their annotations. So let's start with a bit of background here. So ontologies have been widely used as a way to formally structure our knowledge in a biomedical domain. So they have been mainly used for two main purposes. First, they describe relations between concepts within a biomedical domain. As an example, the gene ontology is used to describe relations between different Go terms. And second, ontologies can be used to annotate biological entities. As an example, the gene ontology can be used to annotate proteins and other gene products with specific Go terms by saying that a given protein, for example, has as a function a given Go term. So now a general misconception about ontologies is that they can be entirely represented as a graph. But in fact, an ontology contains a set of axioms which formally describe relations between concepts, and among these axioms, only a subset can be naturally represented in a graph. For example, axioms, subclass relation axioms can be easily represented in a graph, but other types of axioms which are more sophisticated and more complex, such as the ones shown in red here, cannot be, at least not easily be represented in a graph, and they are generally not part of the graph structure of an ontology. So now, given all these pieces of information provided to us by the axioms of an ontology, how can we use this information to perform some data analytic tasks? So the most widely used methods to utilize ontologies for data analysis are semantic similarity measures. So these are basically methods which compute similarity between two classes of an ontology or two sets of classes in an ontology. As an example, the Resnick similarity measure computes similarity between two classes as the information content of their most informative common ancestor. And the semantic similarity measures have already been widely used to perform tasks such as protein-protein interaction prediction or gene-disease association prediction, etc. However, semantic similarity measures have a number of limitations, and I would like to discuss three main limitations which have motivated our work. So the first limitation is that semantic similarity measures treat ontologies as graphs. And as we said, when you treat an ontology as a graph, then there is a large set of axioms which is ignored, and these axioms may encode information which is important and that we want to use. The second limitation is that semantic similarity measures are predefined, so they are based on some assumptions, and they define similarity based on this assumption, and then they are applied the same way on any task or any application and any data. But the definition of similarity may be different depending on the application and the data. So features which we think are important in defining similarity for some applications may not be relevant for other applications and for other types of data. The third limitation is that all we get from a semantic similarity measure is one value, one number, which is the similarity value. And obviously, one single number cannot be very informative and cannot encode a lot of the information provided by the ontology. So the problem we're trying to solve here is the following. Can we define, in a data-driven way, a method to produce feature representations of biomedical entities in such a way that uses all the ontology axioms and all the known annotations between biomedical entities and the ontology? So in this work, we propose Onto2Vec, which is a data-specific method that produces vector representations of both the ontology classes and the biomedical entities annotated with the ontology, and it uses two main pieces of information. First, it uses all of the logical axioms provided by the ontology, and second, it uses the known annotations between biomedical entities and ontology classes. So now let's have a look at how Onto2Vec works. So the first thing Onto2Vec uses are the logical axioms of the ontology. And that would include the axioms which can be represented as a graph and the ones which cannot be represented as a graph, both of them. And then we would apply a reasoner on these axioms to obtain a full set of axioms that describe the ontology. And I will get back to what a reasoner does in just a moment or two. The second piece of information we look at are the annotations between biomedical entities, such as proteins, and the ontology classes. For example, if we know that a specific entity is annotated with a given ontology class, we would express that as an axiom by saying that entity 1, for example, is annotated with or has as function a specific class. So now these two sets of axioms, the logical axioms and the annotation axioms, are merged together within one corpus, and then we would apply Word2Vec on this corpus to obtain vector representations of both the ontology classes and the biomedical entities annotated with the ontology. So now let's go back to what the reasoner does. So the reasoner is used to infer new axioms which correctly describe the ontology, but which have not been included in the original set of axioms. As an example, if we know that C2 is a subclass of C1 and C4 is a subclass of C2, then we can safely infer that C4 is a subclass of C1. So this is a correct axiom, even though it was not included in the original set of axioms of the ontology. So now, given all these pieces, so now let's look at an example of how to apply Onto2Vec. So if we look at the gene ontology, for example, we can take the protein-to-go associations, and we would express all those known protein-to-go annotations as an axiom. For example, we would say that a specific protein has function a specific Go term. So this is our first set of axioms. And then our second set will be the logical axioms provided by the ontology, both the ones in the graph and the ones that cannot be encoded in the graph. So these two sets of axioms together would constitute our corpus, and then we would apply Word2Vec on this corpus to obtain vector representations of both the Go terms and the proteins. As an example, the vector representation of this protein, for example, would look like this. So basically just a bunch of numbers. So now what can we do with these vector representations? So to validate our method, we decided to use our vectors to predict protein-protein interaction on two data sets, human and yeast. And then we compared our method to Resnick semantic similarity and to binary Go vectors. So how to do protein-protein interaction prediction from our vectors? So the first approach we tried, which was a naive approach, was to compute cosine similarity between every pair of protein vectors, and then we would use the similarity value as a confidence score to predict whether two proteins interact or not. Then we obtained our results and we compared them to Resnick. And the results initially showed that our method, which is the yellow curve here, actually does not perform well when using cosine similarity and cannot even overcome Resnick similarity. So we decided to use something which is more powerful and more sophisticated, and that is training a machine learning model on our vectors and then using this model to predict protein-protein interaction. So we trained 3 different models, the SVM, neural network, and logistic regression. And then we did the same thing. We predicted protein-protein interaction and compared to Resnick and other methods. So now the results show that when our model is trained with neural networks and SVM, which are the 2 top curves here, it performs much, much better, and there is a significant improvement over what you can obtain with Resnick, for example. So the AUC values here, for example, show that there is an improvement of about 10% compared to what you can obtain with Resnick. Okay, so far we have proved that our vectors can be used as a data-specific, trainable semantic similarity measures. So now we want to use our vectors to do a task that can never be done with a normal semantic similarity measure, and that is identifying and distinguishing between different types of similarity. So In other words, we do not only want to know whether two proteins interact, but rather what kind of interactions are they having. So we looked at 4 different interactions between proteins: reaction, activation, binding, and catalysis, and we used our trained Onto2Vec model to actually predict specific type of interactions. So the first thing we can see from these results is that Resnick's semantic measure cannot tell you what kind of interaction two proteins are having. It almost performs the same as a random classifier. While our method, especially when trained with SVM and neural network, can to some extent tell you what kind of interaction two proteins are having, which is something that cannot be obtained with a normal semantic similarity measure. Okay, so we have proved that our method can be used as a trainable semantic similarity measure. We have also used it to differentiate between different types of similarity, which is something you cannot do with a normal measure. So now we want to use our vectors to visualize the data and see how they would look like, how biomedical entities would look like together in a 2D space. So we produced the vector representations of 10,000 enzymes, and then we visualized them in 2D space. So when we visualized them, we could see that there were some groups of enzymes that were grouped together in the space. So to understand what these enzymes mean, or what these clusters mean, we decided to color code them by their first enzyme function. And then when we color coded them, we could see that that many of those enzymes that are clustered together actually have the same function. So our vectors can actually be used to identify clusters of entities, such as enzymes, which share the same function and which are within the same functional group. So as a conclusion, we have produced a method that can learn feature vectors of ontology classes and biomedical entities from the ontology axioms and the annotations between ontologies and biomedical entities. And then we proved that our method can be used as a data-specific trainable semantic similarity measure. It can also be used as features to train machine learning models for different tasks. And it can also be used to visualize the data and identify clusters of biomedical entities which are within the same functional group or which share the same function. So at the end of this talk, I would like to thank Professors Robert Hohendorf and Professor Shingal from KAUST, and thanks to all of you for your attention.
So we have quite some time for questions.
I have a question. I'm sorry. Could you explain a little bit more how did you vectorize that? I think it's a protein or that's an ontology term. You showed just a bunch of numbers. How did you generate those numbers?
Okay, so as we said, we use 2 sets of axioms. The first set are the protein-to-go annotations. We express them as axioms. So we say protein X has function Go term something. And then we would also use the logical axioms provided by Go. So all the axioms that describe relations between Go terms. And then we merge these 2 axioms together in one corpus, and we apply Word2Vec on this corpus. So Word2Vec directly gives you vector representations of the words that are in your corpus. So in this case, we are interested in proteins and Go classes. So we would use Word2Vec to obtain vectors for proteins and Go classes. Yeah, this is how the vectors obtain.
I have several questions, but I'll only ask 2. I'll come see your poster or you. So one question is, I find it very difficult to find good axioms in ontologies, in many ontologies. I see that you worked with Go, which only has 2 or 3 types of axioms. So I'm making, I guess, a call for ontology builders here to do include more axioms, because perfect technologies like this, wonderful. And then that brings kind of my second question. There are ontologies, like Bio-Ontology has more complex axioms that are, some of them are nested axioms. So do you ever deal with them, or did you think of ways to deal with them?
Okay, so regarding your first question, you're saying that it is hard to find good axioms to use in the ontology, and that is something that I absolutely agree with. So actually what we have done in a follow-up work was to include the metadata of the ontology, and that is the labels, the descriptions, the synonyms. These are information which are more expressed in natural language and which are more easy to understand. And we actually found out that using the metadata improves the performance much better than what you get with just the axioms. But that is a different piece of work.
Story, yes, I was going to ask, but I was skipping that question.
Yeah, so
that is natural language processing.
That provides much better information. And for the like complex axioms, we actually use all of them, even the ones which are nested, which have like conjunctions, which are negated. We just include all of them in the corpus, and then we leave the task to Onto2Vec to you know, like find what are the relations between you know, like the classes in the axiom.
So you do handle, because
Yeah,
they
just
reason
include them.
differently. Some of them don't make it.
I mean, we don't handle them differently, we just include them.
Yeah. Okay.
But as I said, metadata actually provides much more informative information about the classes and the ontologies.
I will believe it. Yeah. Thank you.
A quick question on the inputs to the Onto2Vec. So the way I understand it, how do the inputs look like into the Onto2Vec in case of, is it just 3 words where it is trying to predict the center word and its neighbors?
Okay, so we actually merge all the axioms in one corpus of text. So I would merge all of these axioms. Each axiom is one sentence. So my corpus will have millions of sentences. And then I feed this corpus directly to Onto2Vec. The
so neighbors of 3.
I mean, not necessarily 3. Like, if you look at some of the axioms, like the 2 last axioms, they are more like complicated. So they do not only include like 3 words. They have like a different. They have like a larger structure, so that every axiom would be a sentence in my corpus, and then the whole corpus is fed to Onto2Vec as a single input.
Okay. Thank you.
So you previously published, or collectively, you previously published a version sort of like this, but used random walks through the network?
Yeah, I think that's a different work.
Right. So I was just going to ask you to compare them. So how do the vectors come out? How are they different?
I think Professor Robert can answer that better than me, because I was not involved in the project.
Okay. Yeah. So this is actually still somewhat work in progress, but the idea is here to have these axioms coming from Go+, where a lot of people have spent a lot of time adding these axioms using CABI, using cell type ontology, using anatomy. And if we do random walks over the ontology structure, it's fine. We can sample some parts of the ontology, but again, we can only sample the parts which we can actually represent in some kind of a graph. And these axioms, so it will not, for example, work. We use OWL RDF XML to represent our OWL axioms. It's very awkward to walk over this, so it's not going to work. So we are going to miss this information. And we see this, that by adding information, for example, links to cell types, links to different tissues, and we don't get this in just the graph structure or just the taxonomy or partonomy, that we can improve this performance. And there we get richer representations, which we now also know how to use. So this will be better if you want to encode for axioms. It will work worse if you have RDF data. Because for RDF data, then it's just a set of facts, and you do not capture this locality anymore. So for ontology axioms, this method works very nice, or better than just having random walks. If you have the data associated, you want to do random walks. And then, yeah.
Thank you.
Any other questions? Okay, so if there are no more questions, let's thank our speaker again.